So one of the goals for you and this class, at least from a teacher perspective, is that you be able to use the experimental design process to do your own experiment later in the year. So we'll have some time to practice starting in the beginning of this year with some lab work. But I think it helps to talk through what's involved in that process and at least have some common language that we can talk about experimental design with. So that's what the set of slides is about. And it would be helpful if you took these notes for yourself and to have that common understanding moving forward. So we go out in the world and we observe things, and that makes us wonder. And our wondering sometimes leads us to questions about the natural world. Sometimes we can look them up or look up the answers. Sometimes we can do a little observational study. Sometimes we want to explore something that could be experimental. And so sometimes you develop an experimental question. So maybe I like feeding birds, and I want to know what bird seed is going to have the greatest diversity of birds coming visiting my feeders. But then maybe I realized that, well, there's not just seed feeders, there's different food types that I want to consider, not just seed. So what differences in bird food will affect the diversity of birds that come to my bird feeders? So sometimes when you start with your question, you refine it a little bit as you're leading into the experiment itself. It's important when you're looking at an experimental question that you are able to identify the independent variable and the dependent variable. So what would be the thing I change in this experiment? I'm changing the bird seed, or the bird food. What am I measuring? That's the dependent variable. Well, I'm measuring how many different kinds of birds come to my feeders. The difference in bird food versus the variable of bird diversity. So an experimental question, you should be able to recognize those two variables within that question. Anybody doing an experiment or having an experimental question is going to want to do a little bit of background research on whatever topic it is they're studying. Now you frame that research in what you already know. So what do I already know about birds? Well, over the years, my parents used black widow sunflower seeds, and it seemed to attract a lot of birds. I've got an established feeding site at home that I've used for a number of years. And I also know this biological principle. More diverse plant communities support more diverse animal communities. In other words, if you have more different types of plants, you're going to have more different types of animals. I also know birds are more active in the morning and early evening. So all of that plays into my experimental design. So what else do I need to know? What are my feed options? I'm not sure. What claims do they make about advertising when they advertise attracting birds? I don't know that either. What benefits might make one food a better choice than another? Well, how easy is it to purchase? How expensive is it? Those things might factor in. And what are my feeder options? So what I know is from my lived and learned previous experience. What I need to know, I can get through online research, a trip to my garage, and maybe a trip to Fleet Farm to look at possible food options and feeder options. Alright, so predictions. That's what hypotheses are. When you develop an experiment, you make predictions about what might happen. So again, have that definition of what a hypothesis is kind of ironed into your brain. So changes in feeder food will affect the diversity of birds feeding at my feeder. That's one outcome that's possible. It's also possible that changes in feeder food will not affect the diversity of birds feeding at my bird feeders. That would be saying that there's no effect of changing the food and the diversity of birds. So experiments are framed with the null hypothesis in mind too. Then, so that was sort of general hypotheses related to the question, but then you might refine your hypotheses based on your specific experimental procedures. Alright, when you are designing an experiment, you change one variable to see if it has an effect on another. You have to try to control for other variables. Keep those constant so that you can rule them out as possible impacts on the thing you're measuring. So for example, I would want to have the time of observation be the same for all the types of food. I would want to keep the type of feeder that I use to hold the food to be the same. Where those feeders are located is going to be important. They need to have a similar type of location, so close to one another. They should be set up at the same level, like the height off the ground, because some birds like to be ground feeders, some are perching feeders. So my experiment needs to control for how high off the ground the feeders are. How much food do I put in the feeder? Because that might impact them, so I have to keep them topped off the same. Perhaps you also have a control group. That's where you would place a feeder out with no food in it, and that would rule out that the birds are attracted to just the feeder. Now, it has nothing to do with food. You start making a list of materials that you think you'll use. So I decided I wanted to have 4 feeders that were identical, 4 different types of food. I wanted a common feeding area, a way to hang the feeder that would be the same, notebook for recording observations, binoculars for seeing the birds more clearly to identify them, maybe a digital camera for recording some observations, and a field guide for bird identification. Now, of course, there's a new tool I learned over the summer called Merlin Bird ID. You can answer your phone. We're going to try that in class. So, a tool for identification. Alright, so that was what I initially thought about. Then added when I was actually doing the experiment. A funnel for adding feed to the feeder so it didn't spill everywhere. I decided that I had a bunch of matching shepherd's hooks for hanging the feeders, and so they would hang the same height and in a similar fashion out in the yard. I ended up with 4 tube feeders that were the same size and color from Fleet Farm, and they weren't too expensive. And then I ended up with my 4 foods: mealworms, cracked corn, traditional seed mix, and a fruit and nut mix. So you make your initial list, then you fine-tune it after you get more details. So your procedures actually change as you're doing in the work, because you refine it, you get more detailed and have more information. Sometimes you modify it. Alright, so this was my initial setup for my experiment. This is at my old house when we lived behind Dollar General. But those are my tube feeders, the four different food types. This was right in front of our kitchen window, so I could sit down and observe pretty easily in the morning and evening. There were shrubs nearby on the west side of the yard, and a basswood tree to the south side. So there was a little bit of nearby shelter birds could go to and feel kind of protected and safe. So what am I not controlling for? Well, there's driveway access over on one side, and then the house and all the stuff that happens up by the house on the other side of the feeders. So maybe north, south, east, west direction is something that needs to be accounted for. And how could I control for it? Well, one way would be to alternate the feeders and have them in different locations around these hooks. Just rotate them daily so that every feeder spends a little time at each of the locations. So then that orientation isn't a factor. So your procedures. When you design your experiment, previous to the experiment and later, you're going to end up with a lab that's going to be more detailed and a little different than what you planned initially. So just be prepared for that. You want to create a document in your procedures that's so detailed that someone else could read your procedures and replicate it exactly as you've described. So sometimes it helps to supplement with diagrams or pictures when you're putting together a procedure. So this shows the different food types. Or like I showed you before, the feeder setup and how it actually looked. It can write that description, but it's also easier if you can see the picture along with it. So what are you looking for? What data am I collecting? Well, I'm going to be looking at the morphology of the birds to help identify them, to distinguish one species from another. But I'm also going to quantify my data. I'm going to keep track of the number of bird visits to each food type per observation time. And then we collect that on a data table. I also am interested in the number of different bird species feeding at each feeder, also on the data table. So what is a data table? We want to make sure we understand what a data table is. Columns and rows, organized with headings, and this is where you collect your data. So oftentimes you refer to this as a spreadsheet. So you will be developing and working with data tables. Usually a data table will have a little title to describe what it's telling you. Here's another data table. On it, I have the independent variable and the dependent variable. Now I've actually put it together this way on purpose. If I put the left column with the independent variable and the right column with the dependent variable, and I have these numbers, I can highlight this on a spreadsheet and put insert graph, and I will end up getting a graph that I can make look like this. And so you will have time to practice making your own computer graphs and data tables. So again, part of the process in doing science. So there's a little bit on experimental design.